Hello, everyone. This is Pocket Size Bio with Dr. Casco. In today's video, we're going to continue Chapter 1 of Molecular Biology of the Cell. This is Section 1.4, Model Organisms, and this is the last section for Chapter 1. And I do apologize for taking such a long time to finish Chapter 1, but let's get to it. So this section talks about model organisms, right? So there's a little bit of introduction, right? Knowledge gained from studying one organism contributes to understanding others due to shared ancestry and fundamental properties conserved through evolution. In this section, the book talks about how different organisms, particularly model organisms, can be used to see some shared traits that we all have in common. And there's, depending on the model organism you use, you can find different types of information, right? Gene function between vertebrate groups, developmental pathways, and practical applications with plants, right? And so forth. So the idea of having these organisms or critical organisms for study about biology is incredibly important, right? And certain organisms are more accessible for laboratory study. Not every organism could be studied so easily. Some attributes of this could be rapid reproduction, right? Maybe they have a relatively fast generation time, or their genome isn't so extensively large, right? Or they just reproduce incredibly fast. So by using these model organisms, it helps scientists understand a more the breadth of what biology is and depth, right? Obtaining more information that is essential for understanding biology. And we could even apply that understanding to human health, right? And human livelihood. Let me just go a little bit down here. Some organisms develop transparently from a fertilized egg to a multicellular organism. That's incredibly important because if you want to if you want to study development, you can see how certain cells are forming in a particular way or they're going through a different cell line, and you can even track that with biotechnology, right? And staining techniques. Cell behavior and tissue formation are these important, concepts, right, to study. So by choosing a particular organism, you can understand, discover new things. Of course, you need to understand how to manipulate the organism. You need to understand what you could learn by manipulating these organisms, right? So you do need to know very important in-depth strategies to understand the cellular processes that you're, of course, going to undertake. Okay. Ultimately, what you're going to embark on and what you're going to research on is largely based on gene function, morphology, right? And so forth. So one of the main things that molecular biologists need to understand is mutations, right? The whole principles of gene function, right? And the whole dogma of biology. In this case, we're going to talk about mutations, reveal the functions of genes. So gene function discovery. Understanding gene functions is essential, right? But it is challenging. Just by knowing the sequence of genomes, it's not enough. Okay. And to understand gene function in itself, you pretty much have to undergo two or have to dive into two approaches that are they're complementary to each other, biochemistry and genetics, right? Biochemistry, it studies purified molecules, it studies protein and DNA, RNA function that is produced by specific genes and tries to examine their chemical activities as a larger whole, right, in signaling pathways in great detail. Biochemistry. And then you have on this other side, you have genetics that study mutant variations of these specific genes within organisms to evaluate and see what are the effects of mutant organisms structure, the organism structure, and the overall function and performance of these genes, right? Basically comparing altered forms with wild types, with the normal variant, with the normal forms and the variants, and so forth. So by having these two approaches in mind, this is a very powerful mean to connect genes and molecules to the cell and the whole organism in itself, right? In terms of structure and function. Here, the book gives an example of an alteration in organism shape resulting from a gene mutation, and this is a um, a study of if I'm saying this correctly, Schizosaccharomyces pombe, a normal yeast, right, compared to a mutant yeast and mutant yeast. And here there's a single gene changing or being mutated, right, from a cigar shape to a T shape mutation. So there's a mutant gene, therefore has a function controlling gene shape, right? And this is what I was talking about, that if you alter the gene, a particular gene, you can understand its function by the consequences, right? By the effects. Okay. So let me continue here. DNA sequence information, utilizing DNA sequence information in molecular biology tools has accelerated process. Sorry about that. Of course, if you know the DNA sequence, right, this enables you to compare um, sequences among different organisms, right? Variations of genes and mutations of genes, right? If you see any differences there, where are the differences, right? How um, um, intense are these mutations, right? But when you compare different organisms, right, you can identify conserved regions within genes. And often these conserved regions or subregions within genes are critical for function if they're conserved among different species. Okay. Mutations created in specific sites within subregions or constructing artificial hybrid genes can help test individual contributions, right, to the gene's function. Okay, so by creating mutations at particular places, you can see what are, of course, the critical areas and also how it alters the protein. Okay, so what are some analytical techniques that are used in molecular biology that you generally would approach? Molecular structure determination, biochemical analysis, right? And cell biology techniques. And as we go forth over these model organisms, we get into some of these in more detail. Ultimately, you want to use these analytical techniques to understand gene function. There is no one technique that you use or no combination of things, of techniques to use. It depends on what you're studying, what your goal is, right? There's no method for discovering a gene's function. Yeah, it is quite of a challenge, right? Um, to undergo. Understanding a gene's function fully necessitates studying its role in the organism's life, not just isolated molecules or cells. Definitely, if you really want to know the gene's function, you can't just study a cell in culture, right? You would need to alter the phenotype in the living organism itself. Okay. Molecular biology began with a spotlight on one bacterium and its viruses. So now we're going to get into the different organisms and how they have helped, right, studying biology over time. The attraction of studying organisms. Learning about particular species makes it more attractive for further study. Each discovery raises new questions and provides new tools for tackling biological questions. Large communities of biologists have dedicate themselves, right, to studying different aspects of the same model organism. Early days, and I'm going to zoom out a little bit. Let me just so you can see the figures more in detail here. Early days of microbiology studies focused a lot on bacterium, particularly E. coli, Escherichia coli. And it was chosen as a model organism because it was very easy to cultivate, right? To culture. Sorry, cultivate. Yeah, it was very easy to culture. It reproduced incredibly fast relatively to other organisms, and it was very adaptable to various conditions. Not all bacteria are easy. Not all bacteria is it easy to culture in a lab, right? So its adaptability to variable conditions. So there's special interest, right, was placed not just on the bacteria, but also viruses that that infect E. coli. Okay, of course, due to their small genomes and ease of analysis. So you had like a two-in-one deal there. You had the bacteria and the viruses that affect the bacteria. Of course, we all know that bacteria, viruses, and those particular viruses that affect or infect bacteria are called bacteriophages or bacteriophages. Right? We all are familiar with viruses. They're small packets of genetic material. They do rely on host machinery to reproduce or replicate, I want to say. Moreover, viruses consist of many families with distinct structures and replication modes. We're going to see these in the later chapters. In the case of bacteriophages, they're viruses that infect bacteria, and they play critical roles in advancing molecular biology. One particular one that is famous in molecular biology is um, bacteriophage lambda and T4 have helped identify and characterize proteins of interest, and they have been used as vehicles to transport genes of interest from one bacterial cell to another, and so forth. Right? Bacteriophages have many close relatives distributed throughout the biosphere, with significant ecological impacts also. So very interesting. You can see let me just zoom in a little bit here, an electron micrograph of a bacteriophage, right, attaching to a E. coli bacteria, right? You can see several bacteriophages, right, pretty much assembling themselves, right, within the host cell, and an even further, right, more in depth, more magnified electron micrograph, you can see a bacteriophage literally on the surface of the cell, where you can see its position is injecting some of its genetic material into the host cell, right? These are cryo-electron microscopes. Okay, very beautiful, actually. All right. Let me just go a little bit downer here. Okay. A role of viruses in gene transfer. So as I mentioned before, viruses can serve as a vehicle, right, as vectors to transfer genes from one cell to another. Okay, so that's when you refer to as a gene transfer there. They can transfer genetic material between cells, right, acquiring new genes, acquiring antibiotic resistant genes, okay, among bacterial species and even among archaeal species. And this is often referred to as horizontal gene transfer, okay, where you're just passing from one cell to another, not necessarily from one parent cell to a daughter cell, dividing, dividing, and dividing. Okay, horizontal gene transfer. It does have some great implications, right? When you get antibiotic resistant bacteria in various environments, because that creates resistance to particular bacteria. And this is detrimental to hospitals, right? Because they become more resistant to medicine and the human body in general. Approximately 18% of all genes in the present day genome of E. coli have been acquired by horizontal transfer from another species within the past 100 million years. So there's has been a lot of horizontal transfer collecting throughout time to what we have now. Okay. Here's a representation of how you the genome, right? This is a way we would represent a genome of bacteria in particular. Let me just move a little bit higher here. Okay, the focus on E. coli as a model organism has accelerated many subsequent discoveries. E. coli K12, this is a laboratory strain that is used a lot, right? Has a genome around 4.6 million nucleotide pairs. And it's of course, we know bacterial DNA is circular. It's a circular DNA molecule, codes for 4,300 different kinds of proteins. Not too much, not many compared to eukaryotic genomes, right? So this E. coli is one of the most extensively studied organisms, right? It's given us many, many insights on how the mechanisms of life and how a cell regulates itself. There's been many basic genetic mechanisms discovered in E. coli, highly conserved throughout evolution, shared with other organisms among eukaryotes, including us, right? Um, so it definitely sheds light on a lot of biomolecular activity, right? In terms of genetic diversity, different strains of E. coli exist, right? Despite belonging to the same species, there are significant genetic diversity, quite difficult to see just on their morphology, right? Um, Hundreds of genes that are different between strains, okay? But nevertheless, you have this idea of horizontal genes transfer, common in between archaeo and bacterial species, can contribute, to the genetic diversity of E. coli and other bacteria also. Okay, so that's in terms of bacteria. Again, as a model organism, we go to the next organism, which is yeast. Okay, serves as a minimal model eukaryote. So now we're jumping from prokaryotes, right, into eukaryotic organisms. Let me just move a little bit higher here. And so, yes, there has been many discoveries using bacteria, but scientists want to continue discovering with cells that are more closer or closely related to us or have the same build as us, and as in the case of eukaryotes, right? So there's a need for model organisms in eukaryotic studies, right? The molecular and genetic complexity of organisms necessitates on focusing resources on selected model organisms to understand that complexity, right? So, selection of Saccharomyces cerevisiae as a model eukaryote. We know this organism as yeast, right? In terms of one of the most minimalist model organisms for studying eukaryotic processes, it is a single cell species, right? Does have an ideal simplicity for analyzing, internal, of course, cellular mechanisms, molecular mechanisms, without needing to really have the difficulty of multicellularity, right? So it's a single cell that you can study, but is also an eukaryote. Okay. So some characteristics of S. cerevisiae. I do apologize. I am aware of the italics that I should be having here. My apologies. Um, Again, Cerevisiae is a small single-cell member of the fungal kingdom, right? Genomically is more closely related to animals than plants. It possesses a cell wall made out of chitin, right? It does have mitochondria, of course. It lacks chloroplast. Okay, so it's more similar to plants, sorry, to animals than plants. Under favorable conditions, it reproduces every 100 minutes, either vegetatively or sexually. So it could sexually reproduce, or it can asexually reproduce, right? Of course, here's an example of yeast. You have these little electron micrograph there, surface electron micrograph, right? And a transmission electron micrograph there, where you can see all the organelles. Let me just zoom in here, actually. Hopefully it clears better. You can see the nucleus there very nicely, all the ribosomes embedded throughout the cytoplasm, and so forth and so forth. Okay. Much, much more complex than prokaryotes. I'll leave that a little bit there. Okay. Again, so you have this yeast genome, though small, it is quite small for a single cell organism. Okay. It does perform all the necessary mechanisms as a eukaryote, right? Being an eukaryote. This organism has been extensively studied. Mutants have been made and are available for literally every gene within yeast. So you can apply, right, and create studies to observe any missing gene, right, under different environmental conditions that you want to study, right? So advantages of Saccharomyces cerevisiae for genetic studies, right? Its small genome does facilitate high throughput studies of gene functions. Okay. Extensive research has been done over 50 years, elucidating crucial eukaryotic processes like cell cycle division, meiosis, right? Cell division, sorry, cell division cycle, right? Meiosis, chromosome structure, nuclear organization, gene expression mechanisms, organelle formation, and protein secretion. A lot of understanding of cell biology has also come from yeast, right? Many fundamental processes in yeast closely resemble those in human cells. So, insights to human biology has been discovered because of yeast. Okay, let me just take a look here. All right, let's go to the next little subtopic here. The expression levels of all genes of an organism can be determined. Okay, genomic characteristics of S. cerevisiae, the complete genome sequence of yeast comprises of only approximately 12.5 million nucleotide base pairs, including mitochondrial DNA. So it's only 2.7 times larger than E. coli, so not that big. It codes for 1.5 times as many distinct proteins as E. coli. So definitely not an incredible difference compared to other genomes in the eukaryotic realm, right? So it's a good way to compare genome analysis, right? Despite being an eukaryote, genome size and protein coding capacity is quite similar to bacteria like E. coli. Let me just go a little bit here down. Similar selection pressures, such as the need for rapid proliferation, have likely influenced the yeast's compact genome, right? Since you need to reproduce quickly, the likelihood of having a not so large genome is higher, right? Because you need to survive instead of spending time replicating more. Okay. So, significance of complete genome sequencing, right? Knowledge of a complete genome sequence of any organism, including yeast, offers new insights into cellular mechanisms, just as I mentioned before. We're going to see techniques a lot later in chapter 8 and down um, that enables simultaneous monitoring of mRNA production from every gene in the yeast genome under various conditions. Real-time analysis can reveal changes in gene pattern activities in response to changing conditions, right? So these analysis can be used to can be extended to mutant cells lacking specific genes, so you can see the consequences of or the influences of these environmental conditions, right? Um, on that particular genes and how it influences other genes also. Okay, so the impact of genome sequencing on gene expression studies. Um, Using yeast, right? These are pioneer approaches can facilitate the understanding of gene expression, control mechanisms, or control systems in any organism with a known genome sequence and genetic manipulability. Okay. One of these going to into another organism right? Arabidopsis has been chosen as a model plant. So we're going from fungi in to plants, focusing on plants now. Evolutionary relationships among multicellular organisms. So despite the vast diversity among plants and animals, they share closer evolutionary origins and basic cell biology compared to microscopic single-celled organisms. Okay? Bacteria and archaea, their evolutionary lines kind of diverged approximately 3.5 billion years ago. Okay, billion years of evolution. Vertebrates and insects separated about 700 million years ago. Fish and mammals 450 million years ago. Different species of flowering plants, a lot more younger, right? About 150 million years ago. Nevertheless, you could get insights of these model organisms and shared characteristics. So you have evolutionary relationships among all flowering plants, right? Which would allow insight into the cell and um, uh, yes, and molecular biology within this group of organisms. Just by studying a couple of plants, you can see many, many shared characteristics. Okay. And in the case of plants, you have this model, right? Arabidopsis thaliana, a small weed in the cabbage family out of nearly 400,000 known species of flowering plants. And this is a little visual of how this plant looks like, okay, for plant molecular genetics. Okay. Arabidopsis A. thaliana can be cultivated indoors, very, very large numbers, produces thousands of offspring per plant every 8 to 10 weeks. So that's quite fast compared to some other plants. Okay. The total genome of Arabidopsis is approximately 135 million nucleotide pairs, 10 times, about 10 times the size of the yeast genome. Contributions of studying Arabidopsis. Okay, definitely some key features of plants, mechanisms of underlying flower development, coordination with seasons, phototropisms, and the ability um, eh, to ability to grow towards sunlight. Okay, or any stimuli for that matter. I wanted to say. Okay? Cell-to-cell signaling via hormones, right? Plants' innate immune system against pathogens. Some quite insightful things in terms of plant research. Okay. Comparative developmental programs have been established due to these insight of these model organisms, right? Between plants and animals has revealed common principles, offering insights into evolutionary transitions from single cell ancestors to large, highly differentiated multicellular organisms. Okay, so we go into animal cells um, from model plants. Okay, the world of animal cells is mainly represented by a worm, a fly, a fish, a mouse, and a human. Interestingly, the distribution of biomass and species, plants constitute 80% of the biomass on Earth, whereas animals make up less than 0.4%. It's terrible. Despite this, animals represent the majority of all named species, right, of living organisms and are the most intensively studied group. So, among these model organisms, you have these five. You're going to see these throughout this whole book being used. Um, Five species have emerged as model organisms for, of course, molecular cell developmental studies. Um, the simplest one, if I can say, Cyanorhabditis elegans, C. elegans, as you can see here in this panel, in this visual here. Drosophila melanogaster, Danio musculus, Homo sapiens, right? So we have this eelworm, you have the fruit fly, you have the zebrafish, you have the mouse, and you have humans. So C. elegans, we see here, let me just zoom out a little bit so you can see better. Very small, harmful harmless, harmless relative of the earworm. Very short life cycles and can survive in suspended animation in freezers. So this is very useful for research, right? Indefinitely, right? It's a very simple body plan, right? So you can use this for genetic studies. Many research laboratories do use this to today. Organism develops with precise regularity from a fertilized egg cell into an adult worm consisting of exactly 959 body cells, if you include the egg and sperm cells. Quite interesting, right? Its genome, about 100 million nucleotide base pairs, which codes for 20,000, approximately 20,000 proteins. Okay. And you see a little visual here. Even itself, you can see some of the intricacies inside its body. Okay. Contributions of C. elegans. So C. elegans serves as a model for many developmental and cell biological processes, including programmed cell death, or the idea of apoptosis was found by this model. RNA interference, or RNAi, neuronal connectivity, right? And other aspects of cell biology have been discovered because of studying C. elegans. Okay. We move into Drosophila. Drosophila melanogaster, the fruit fly, has been and it still is a model organism for genetic studies for animals for a great period of time. Pivotal role for the foundation of classical genetics, right? So, uh, one thing that's quite special about Drosophila melanogaster is its giant chromosomes that you can see here. Okay. They have a characteristic band appearance that helps facilitate, and you can quantify and tag and kind of monitor, right? How this hereditary information is undergoing, is moving from cell to cell, chromosome behavior essentially. So mutant flies provide evidence correlating specific changes in heredity with the alterations of these giant chromosomes. Okay, and here you can see some examples of some mutations that have been done in the laboratory. Here you have development of a normal fruit fly, okay, where you have the antenna have been replaced by misplaced legs. Okay. All right, let me continue here. So contributions to understand development, Drosophila demonstrated how to trace genetic instructions to the structure of an adult multicellular body, right? So that's quite a breakthrough when you're having multicellular organisms, right? By having mutant Drosophila with misplaced or mispatterned body parts, that enabled scientists to identify and characterize genes within a larger scope of DNA, right? Characterizing genes essential for proper body development. So, identification of Drosophila genes led to the discovery of homologous genes in vertebrates. Okay, so not just development of the fly, but also developmental genes that many vertebrates have, right? Revealing similarities in molecular mechanisms governing development. Okay. Advantages of using Drosophila, right? Rapid development. Drosophila progresses from a fertilized egg to an adult in only 9 days. So that's incredibly important when you want to evaluate development, right? Cost effectiveness, right, and ease of breeding compared to vertebrates is relatively low, incredibly low. Smaller genome size, approximately 14,000 protein coding genes out of a genome nucleotide base pair sequence of 180 million. Okay. Availability of mutants essentially for any gene. So this is incredibly important because you have a whole reference genome that you can work with, right? The scientists have established over a long period of time. So continued insights and research on Drosophila right, extends beyond developmental genetics to uncovering insights into various biological mechanisms, including pathogen neutralization. If you apply a pathogen to the fruit fly and see the defense mechanisms, how what are the consequences, right? What are the effects of different forms of treatment there? Brain processing of environmental stimuli. Okay, so that's the fruit fly. Let's dive into amphibians and fish. Okay, let me zoom out just to make sure that I'm showing you all the visuals there. Okay, so for the frog and the zebrafish provide highly accessible vertebrate models. Okay, talking first about this Xenopus laevis, the African clawed frog, used to study early development, right? Embryonic development in vertebrates. Okay, so now we're kind of moving into this vertebrate field. Large, they're very easy to manipulate the eggs, right? They have relatively large eggs to manipulate in the laboratory. They're fertilized outside the animal. It does facilitate the whole idea of sexual reproduction and development, embryonic development. Okay. Poorly suited to genetic studies, though, cytoplasm isolated from the eggs allow biochemical studies of fundamental processes like cell division to be studied. So even though genetic analysis might be a lot more difficult for amphibians, right? They don't mention here the newts and salamanders that have incredibly large genomes, but they have regenerative capabilities, right? Um, they are used a lot in studies relating to tissue regeneration, right? Like the axolotl or Synops pyrogaster, the Japanese fiber-bellied newt. Okay, so yes, for development, Xenopus laevis is a great animal to work with. Okay, and then you have Daniel Rerio, the zebrafish, well suited for genetic studies. It does have a compact genome size, half as big as that as the mouse and a human. All right, we're getting closer in that evolutionary tree in terms of similarities. It does have a short generation time, about 3 months compared to Xenopus. Mutants, many mutants are available, and manipulation of genes and genetics in general is relatively simple. Here's an example of a zebrafish here. One of the most important things is that the transparency of the eggs and the development of these organisms' lives is transparent. So it does help in observation, right? Easy observation there. Sorry. Tissue and cell behavior during development. Okay, so it's a crucial model for understanding the heart and circulatory system, especially if you're doing studies relating to regeneration. The fish does have some ability to regenerate. You can cut approximately 20-30% of its heart in the lower um, area of the heart, and it's able to regenerate it. Okay, so definitely for tissue analysis and processes like regeneration is important. Okay, so these models, through different characteristics and suitability for genetic analysis, they do play essential roles in studying vertebrate development and some fundamental processes. Okay, especially when you compare it to humans. Okay, we move from the fish to, let me just zoom out, to the mouse. Okay. The mouse is the predominant mammalian model organisms. Okay, so having a mouse, having a vertebrate mammalian model is fundamental to understanding human development and um, uh, human research, right? Uniformity of mammals. So mammals exhibit a great deal of uniformity in terms of genome size, function, cell biology, protein functions, right? Molecular mechanisms are quite uniform, if I can say. There are anatomical differences, right? That amongst, of course, we see all the diversity of life among mammals primarily relate to size and proportion rather than fundamental structure. Right? It's a very nice sentence there, right? Genetic resemblance among mammalian species, they so mammals typically possess 1.5 times more protein coding genes than Drosophila. 1.5 times. Okay. Mammalian genomes are 16 times larger than those of Drosophila, right? In comparison of nucleotide sequences or amino acid sequences for that matter, corresponding genes show high similarity among mammalian species. So you do have some good conservation there. Okay, high similarity. Over 80% of amino acids are usually identical when comparing orthologous proteins between humans and elephants. So the book is trying to portray a good um, uh, um, idea, right, of this concept of similarity between mammals. Okay, here you see a nice little panel here. We have percent amino acids identical in, for example, hemoglobin chains, right? So times of divergence of different vertebrates, and here you have this scale that is to the left that estimates timing, right, and geological era from last common ancestors of each specified pair of animals. Right. Each estimate is based on comparisons of the amino acid sequences of orthologous proteins. The longer the animals, the longer the animals of a pair have had to evolve independently, the smaller the percentage of amino acids that remain identical. So of course, so the longer they evolve independently, the smaller the percentage is identical. Okay. So when you compare, for example, the human and the chimp, that they have evolutionary divergence, and when you have this timeline here, millions of years, right? Human and the chimp have almost 100% amino acids identical. Human and orangutan, 98%, right? So let's go to one that's not so much. Bird and crocodiles, 76% identical in terms of hemoglobin. If you see as we go back in time more, humans and sharks, our percent comparison to fish to these sharks, 51% amino acids are identical. Okay, so you see over time, as we go back in time, we tend to have less similarity. Okay. All right. So mouse as the model organism here. Yes, mouse is favored as a model organism studying mammalian molecular cell biology. There are naturally occurring mutations in mice that do offer that or do mimic human mutations. Okay. Methods have been developed to create mutations in mouse genes that could be used for functional testing or of genome regions, right? Mutant mice provide valuable insights into gene functions across various cells or cell types in the body. So, mammalian studies are fundamental to understanding, mouse studies. If I've been saying mammalian, it's mouse studies are fundamental to understanding mammalian biology and are relevant across multiple chapters in biology textbooks. Right? Very nice example here. Similar mutations produce the same effect in human and mice. The human baby and a mouse shown here have remarkably similar abnormal white patches on their foreheads as a result of a mutation in the same gene called KIT. Okay, very nice example here, right, where you see these white little patches, which is required for the normal development, migration, and survival of some skin pigment cells. Very, very interesting there. Very nice picture. Okay. Humans are unique in reporting their own uh, peculiarities. Sorry about that. The human genome as a model. So humans offer special interest in genetic studies due to the documentation. Of course, we're living organisms, we're rational living organisms that we document our everything we've done through time. Due to documentation of genetic disorders and medical examinations, human population we have a vast size, self-reporting, enables the cataloging of mutations and their effects, right? So, of course, studies. Human genome consisting over 3 billion base pairs has been sequenced for hundreds of thousands of individuals. So many individuals, which facilitates the kind of sequencing, the identification of any genetic change. I don't want to say any, maybe we haven't 100% known every phenotype responsible for various phenotypes, though. Okay, we do have a complex genome, right? It is incredibly complex, and it does reflect the degree of variation and variant genes that occur in the human population. On average, any two individuals differ at roughly 4 million sites in their DNA, in their sequence of DNA. Okay, so understanding genetic variation helps explain disease susceptibility, drug responses, right? Insights to human history, including population movements, interbreeding, infection, and diets. We're going to see all these things in particular later on in the book. Okay. Oops. Yes, later on in the book. There you go. So, utilizing model organisms, right? Insights from various model organisms, including human, mice, fish, flies, worms, and so forth, helps our overall understanding of genetics, right? It doesn't necessarily mean, since we have higher order organisms, that we're not going to use these different types of organisms. It does depend on what study you're going to do. DNA sequence similarities between model organisms help map corresponding and enhances correspondences and enhance overall comprehension of genomes, right? So to understand cells and organisms will require mathematics, computational computers, um, and quantitative information. So it's quite interdisciplinary, if I can say. Understanding cellular function, of course, does have challenges, right? There is a lot of data, especially nowadays with sequencing, high throughput sequencing, single cell sequencing. So, requires advanced analytical methods, more powerful computers, right? Understanding how cell works does remain a significant challenge as as technology becomes more effective. That is a new pool of data, new pool of results, new pool of information that we need to analyze. So, um, biological systems are characterized by feedback loops, right? Making their behavior difficult to predict intuitively. Transitioning from qualitative descriptions to quantitative understanding requires a lot of computational power, mathematics, quantitative information. But who knows? Maybe we're diving into this whole AI era. Maybe that does facilitate our and our research, right? Our discoveries a lot faster. Only time will tell, right? Limitations to current knowledge. This is probably a little bit outdated from what I did already. Present knowledge of cellular systems is incomplete. It is filled with gaps, uncertainties, and qualitative descriptions. Cell biologists often represent their understanding through diagrams rather than quantitative models. Progressing from qualitative to quantitative descriptions, it is a major challenge in contemporary cell biology, right? So, addressing the challenge, okay? Progress has been made in understanding simple fragments of cellular machinery involving a few proteins and genes. But again, new approaches, we're going to see in chapter 8 some of the more novel ones, and yet that probably this is already outdated, um, to address increasingly complex questions in biology and implications of knowledge and understanding. Knowledge and understanding empowers interventions to prevent and treat disease, improve crops, control microorganisms for various purposes. So, molecular biologists, right, their ability to decipher genetic information is transforming humanity's relationship with the living world. Okay, so that's quite interesting there. Understanding cell biology equips individuals to contribute to the ongoing biosciences adventure of the 21st century. Okay, so I do apologize for not finishing